So in the last video, which was a part 2 of cellular development, we learned about DNA replication. So we finished that topic. But now we're going to be learning about protein synthesis, which is another pretty complicated topic, and I'll try to make it as simplest as possible for beginners like you to learn about biology. So let's go. I didn't really find a picture, so all this will work on drawings. But this is also a pretty easy understanding of this. Okay, so we'll be learning about protein synthesis. Protein synthesis takes place around the whole cell. So that's why we will be taking the whole cell as an example. So we have the nucleus over here, these small and basic drawing. I do not want to draw all the other stuff around it. So that's why I'm only going to draw the three main organelles in this process. It's really messy. These spikes here, it's not so accurate, but these are the nuclear pores. If you looked at the part 2 um, of the anatomy of the cell made by Genius Foster, you understand what the nuclear pores are. But I'll just say it roughly. There are holes in the nucleus which allows things to both come in and out of the nucleus. So it's not so good, and it's not such a good drawing, but it'll help you understand. Then the other important organelle is the Golgi apparatus, which is other organelle all the way over here. And finally, we have ribosomes, which are these other tiny moving organelles around the whole organism. And I'll just draw a small portion of the endoplasmic reticulum the rough endoplasmic reticulum here. Okay, so this is a pretty basic drawing, and now we're going to go inside the nucleus. So if you were to go inside the nucleus, it would look something like that. So this one's going to be a neater drawing of the nucleus. This is the nucleus right now I'm drawing. Okay. And inside the nucleus, we have, if you watch the part 2 of cellular development, you will have learned DNA replication. So DNA replication, in the leading strand and lagging strand, the primase made an RNA primer, which was then removed by the DNA polymerase, because then it was no use after all the genetic material was transported to the other RNA primers. So then after it's removed, after getting all the genetic material needed to, when getting genetic material, it gets genetic material for the G and T or C and A DNA strands, which you have learned in the DNA replication video if you have watched that. After removing these RNA, after removing this RNA, this is called the RNA messenger. It's a simple term. So then this RNA messenger, after knowing both giving the genetic material to the C and GT DNA strands, it also took the genetic material needed to make protein for the cell. So now it knows how to make protein. So then what it does, it goes swirly, 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 going through the nuclear pores and coming out. So now it's outside and into the cytoplasm of the nucleus. Then it comes in the endoplasmic reticulum, where organelles called ribosomes are just sticking and waiting over here for RNA messengers. This happens a lot, but I'm only going to be using one of the examples. It happens a lot in the cells. So then what happens is a ribosome, what it does is it takes this RNA messenger and it uses its messages and genetic material to help make what we call protein. So to make protein, you need amino acid. So on the RNA, there's already some amino acid which was used to stick the RNA to the DNA. And the ribosome collects all this amino acid. Okay? And after it takes all the amino acid and all the genetic material about how to make the protein, the RNA floats back into the nucleus and is used for DNA. So then after that, what the ribosome does is then it takes these amino acids and it transports itself across the nucleus and to the Golgi apparatus. Here, the Golgi apparatus, then it takes all the amino acid and it makes it into a more productive shape, which can be, it makes its amino acids, constructs it. So bad vocabulary, whatever. So what it does is it constructs this amino acid and makes it into a shape which can be more useful for protein. So what it will do is it will make this amino acid into a more useful way and then release it. And this useful thing here, which it just made, is protein. And this just floats around the cell, and then when an organelle needs it to make something, or if it needs it for any reason, it will just use the protein. Otherwise, and then this is technically how protein is made inside the cell. So I hope this has helped you in a way of understanding how protein is made. And uh, as we end this video, I hope you like and subscribe this video, share it to more of your friends and family for more videos, and see you guys soon. Bye bye.